Good morning, everybody. So I'm the only one standing between you and your lunch. I'm not gonna make it long. My name is Reem Aboukhaddour. I'm the serial pathologist working at AFC Lethbridge right there somewhere. And today my helper is Dr. John Laurie, who is a serial biotechnologist, joined us recently from Cambridge. So here I'm gonna give you the task of helping me, please. And there are numbers. On the agenda, I was supposed to talk about leaf rust and stripe rust and leaf spots. I'm gonna touch on those, but I'm not gonna expand too much because I realized this summer people so much send me samples, and they're asking what is that. And I thought, okay, maybe I dedicate some of my part to explain what is that. So I'm gonna start by giving you some tips of diagnosing, how to diagnose a disease, and some of the conflicting diagnoses, sometimes how you could confuse one symptoms for so many things. I have to admit, when people send me, especially when the cereal crop at the early stage, and I work with wheat, so most of my examples are wheat. They usually send me images like that. You see there is yellowing in the plant, and they ask, people who are far, just come closer, I cannot move too much. So they are asking, what's wrong with my plant? Is it disease? It's nutrient deficiency? What is that? And they ask the expert, and my reply usually, wow, that looks not normal. But well, you cannot really judge from one pick what is wrong with your crop. And I have some scenarios, and thank God we have a great pathologist around. So I got help from, Kelly Tarkenton. He shared with me some of his scenarios, images today I'm sharing. So in this case, the plants look at the early stage, yellowing leaves. And you might suspect that either nitrogen deficiency or seed blight or some of the diseases. What you do, first you have to look at your whole crop. You look at your whole field. You have to ask this question. First, the history of what you were growing before the season. Then you ask, is this problem restricted to certain areas, certain lines, or it's spotty, it is random, does it have a pattern? That will give you a clue whether it is abiotech or biotech. But then you're gonna look at the whole plant, because you're gonna dig that plant and compare the roots. In this case, if you look carefully, it was actually a problem all over the field except the header lines. They did have normal looking plants over there, here. So turned out that this was a problem of seed bedding. The seed bedding was really bad, and the header plants were okay because when the track turned, double the seed and double the fertilizer. So the contact between the seed and the soil was so poor, so the plants strive for nitrogen deficiency. So it was a case of nitrogen deficiency, and you could actually look at the root. The yellowing one has a bit less developed root compared to the healthy one. And now, if you've seen that, you can rotate this. Actually, you guys, you can just spread it around. Okay, what was that scenario? Okay, here is another scenario. So here, you could see in the field, there are a spot, and this is also, I borrowed that image from Kelly Tarkenton. There is a spot of the field where the plants are yellowing. Now, if I look at that and I'm plant pathologist, I say, wow, that's not abiotech, it's only spots, and the neighboring plants look perfectly fine. But then, you look closer, you find, and this is not the great focus photo, you find volunteer canola surviving very well. And the plants, if somebody send me those plants, I will suspect nitrogen deficiency. It wasn't actually disease because there was no color on the seedling or the roots or anything. But then it turned out that this was a spill of chemical. And that's, so that's how you cannot really judge from one image, and I cannot emphasize enough looking into your whole field and your whole plant. And no, I actually can share this again. Now, this is what look like a disease, and this is either seed blights or like a seed decay and causing seedling blight. So those type of diseases are caused by several pathogens, Fusarium species, Rhizoctonia, Pythium. It's very hard to tell which one, and it's very complex, but so we can call them like a issue of damping off or seedling blights altogether that cause like yeah, maybe. And yeah, so like it caused this, what do you call it, brown, black color, either right above the ground at the crown or under in certain cases. And you are right, I'm gonna mention something here. Well, you know, I gave you a lot.
Thank you.
So, what I was gonna say. So those early stage crop diseases are really hard, the most hard to distinguish because they are affected by so much factors, whether it's nutrient or whether it has all to do with the good establishment of the crop at the beginning stage. So preparing the seed bed, analyzing what you have, fertility in your soil, and I cannot emphasize more than enough. This is the only time I love fungicide when it is on the fungicide and insecticide on the seed. It's so important to have high quality seed. I cannot emphasize that more than enough. Now talking about stripe rust, I'm not gonna touch too much about it because you guys, it's easy to diagnose. You know what is stripe rust? Who doesn't know what is stripe rust? See? So I don't need to talk about it. It's this yellow bustule all along the leaf. And to control it, it's one of the most challenging pathogen to control. You cannot really, like with other diseases, you can go away with rotation, managing your stubble. These things blowing thousands of kilometers and can rain shower over your crop. So the best effective way actually having genetic resistance or fungicide, let's be realistic. So genetic resistance does work, and yeah, shame on the breeder, never show you that. I stole that from their plots. So this is an image of standing side by side, susceptible and resistant line. So.
Did you say that was Harpinder's plot?
I'm not saying. Yeah, somewhere. And resistance does work. Why? Because you might have a little bit of a stripe, but you don't have the build-up of inoculum on that. So the danger of the pathogen building up and spreading is really not much. So you might end up not needing to spray much when you have a resistance compared to susceptible.
Would you say that stripe rust, though, is one of the more challenging in the sense that it's always evolving?
Yes.
Not as stable as, say, leaf rust or
you know, all rust evolve. Is not, for example, stripe rust, rust in general, rust pathogens can change virulence very fast. With tan spot, we know for a long time we have in Canada race 1 and race 2. But for stripe rust, it's always giving you different races because it just mutate fast. It just has that ability to generate so much virulence. It's more of explosive disease.
I should have asked Harpender earlier. He's not catching my voice, but are the new ones, when they say they're resistant to stripe rust, have they got 2 or 3 races built in?
We try to screen to what we get the pathogen. Like we update our resistance to build up on what is coming in new.
Okay.
Yeah, but you can ask him that question.
The problem is it's resistant till it's not. So one year you had a variety that wasn't, you know, that showed good resistance, and you find a little bit in it the next year, and you're like, oh yeah, you're good. And all of a sudden, well, the resistance failed in that variety over the winter, and so you got a field full of stripe rust.
So you're a proponent of the certified seed program then? Keep refreshing your.
Yeah, that's why we keep breeding for. That's why that's why the research is always one step ahead.
So what I'm saying is it's not. You guys say it's very clear. Oh, this is resistant. Don't spray. It's not that clear in the field.
No, it's not. But that's why you scout. For me, as a look, if you go in your field and you find your plants at early stage has rust on them, you might want to prepare for spray more than once, because at the end you want to protect your flag leaf and your ear. That's contribute more than 65% of your yield. But if you scout and you don't find much, then why you want to spend 100,000 on fungicide, right? So that's why it's so important to scout. Now, to be honest with you, I am not a farmer, and I feel pain for the farmers because they have this huge field that I don't know how they scout. I hope sometimes technology will help, drones or something, but that might come into play.
But my point was keep up with the newer.
I'm gonna,
as she said, they're
yeah,
putting the new ones in.
I'm sorry, I'm gonna cut because I wanna move on something else, and I'm sorry, but we can talk later for sure. What I'm gonna talk about is the other confusing diseases when you get this. I get a lot of people asking me, is this Fusarium head blight? Well, there are a lot of things that can cause the blighting of the head. And I'm sure you guys heard of root rot, take all. So this kind of typical system of symptoms of root rot. Now with Fusarium head blight, actually it happened, the infection happened at the anthesis and starts somewhere in the middle of the spikelet, and then it might go up and down, and you might see the whole spikelet as well kind of blighted. But then you have just the stem right under the head also sometimes turn to that brownish purple color, and then sometimes you find that pinkish color also, that indication of Fusarium head blight. Fusarium head blight really like a certain combination of wet and hot weather. So if you take all that in consideration, you might be able to see, oh, is it Fusarium or not? Besides, Fusarium does occur like with take all and root rot, because it's a soil-borne diseases. It happened in certain spots in the field. Fusarium can be spread all over. You will see the spikelet head all over in certain time at the anthesis. So these are a few things to tell. Besides, I got this sample actually sent to me, and then I look at the head, it's blighted. Then I look at the rooting, there was a pinkish, that was root rot. With take all, you get that blackish color under the ground and above the ground kind of area. And at the end, you have that, what they call it, the sooty mold, the black mold. So I'm gonna share that with you. These are few tips. At the end, when suspecting Fusarium head blight, test in the lab is the way to rule it out or in. Do we have more time, or should we go for lunch?
Maybe 5 minutes.
Okay, the other confusing things, and I work a lot on leaf spots, and this is an image of, what do you think this? Oh, well. Yeah, well, it's you guys.
Put it on your head.
Even me, I work on tan spot. I find that very hard to distinguish the symptoms because it depends on the stage of your crop, depend on whether the progress of infection. The typical infection, we have some clue to tell which spot is which. We have mainly 3 leaf spots in Canada. We have the tan spot in Western Canada. We have Stagona spara nodorum, and we have Septoria, Septoria triticum. There are few clues to distinguish between them. With tan spot, it has this oval lesion. There is a dark spot in the center and surrounded by chlorotic margin. Now, leaf spots get with the other ones, they have different distinguish. I'm gonna emphasize on tan spot because I think it's the most important one, and we might be running out of time. With the Septoria, you find the black structure, the pycnidia on the lesion, and the lesion more of long shape kind of looking. I'm gonna distribute images. People a lot of the time, especially breeders actually, they come with this physiological leaf spotting and they're asking me, we have leaf spots on our, I was like, no. How to distinguish leaf spot that is caused by physiological damage versus pathogen. Pathogen tend to be normal. Doesn't have a specific pattern. Even in the field, you will find it in certain area more than other area, and so it's not gonna be like a. In this case, I went to field last year and I was so happy. I thought I got tan spot, I love that thing. And I started to pulling the leaf and telling my technician, this is leaf spot. And he was like, not very convinced, I guess. There was a pattern of yellow and green, yellow and green. Took the samples, nothing grew out from them. It was physiological spots for some reaction of heat or light, I don't know. But then when we went back to that field, it was totally healthy. So what I meant, physiological leaf spots doesn't happen in random, they have a patterns. Even on the leaf, they are more of pattern. They don't progress, disease always get worse. So, and there is a progression within the canopy. You get your lower leaf hit so bad and they start to infect the upper canopy. So there is a progression in the disease that does not happen with physiological spots. And yeah, and I think, do you want to hear about streak mosaic virus or should we go for lunch?
We're not died anyways. Go ahead.
All right. So, well, I'm not gonna make it long, and I lost my streak virus.
Oh, I just passed it.
Did you? Okay, that's fine. Yes. So streak virus, I got a lot of samples this year, people asking about this, and actually we test a lot of them to be turned positive. It's a viral disease that is transmitted by a mite. This mite you cannot see by the eye, it's very small, and it doesn't have a wing. But it depends on the wind for its movement. So you need a microscope to see that ugly four-legged worm-like animal. And it causes this streaking on the leaves, yellow and green, and that's why it's called streak mosaic virus. And the mite actually is called the curl wheat mite. It causes the leaf to curl up, inward and up. And it does do its feeding. It like to keep in that area where it's curled because it's kind of cool and wet for it. And then when the mite done feeding, it climb up to the upper head and kind of hang by its tail waiting for the wind to carry it over. This thing can carry by the wind about 4 or 5 kilometers and cause the infection. Within 30 minutes from feeding on other plant can transmit the virus. There is no chemical control for it. There is no effective chemical against the mite. You cannot treat virus with chemical. So the only way is actually to not provide it with a host. This thing survives in the winter more than any hardiest winter, but it cannot survive on a dead tissue. So it needs that tissue to be alive to survive. So there are certain culture way you can maneuver around to prevent infection. So if you have volunteer wheat, and it can infect actually wheat, it causes more damage on wheat, whether it's a spring or winter, it causes more damage on wheat. It can also infect other cereal like barley, oats, other grasses. So if you have, for a case, your field full of volunteer wheat and the mite and the virus surviving there, and you sow your crop, and your crop probably survive, if it is winter, these things survive the winter. Early in the season, when it infects, it causes the most damage when it infects the crop at the early stage. So what we call that, the green bridge, because it allows it to survive from winter to spring. So breaking the bridge, whether it was through the winter to spring or whether it was from winter wheat in the spring to spring wheat, that will probably help. There are some resistance. We work on resistance actually, and hopefully I get the project I put for this thing, and we're trying to incorporate the resistance for mite and the resistance for the virus. And this is typical problem to South Alberta. Has been as long as the 50, recognized, even in part of Saskatchewan. And I think I'm gonna stop. I'll let you guys go for your lunch. So, any question?
So if you've broken the bridge, where did it still come from? The mites.
You know what? If you break the bridge, you most likely will get it less. If it came from neighboring field or it's blowing, but
It will blow in.
Yeah, and you know something, it's not only like you have to make sure that your stubble is totally dead, no green tissue. It would survive. Otherwise, it would survive. But if you break the bridge, you will less likely to get it unless your neighbor has it.
Join me in thanking Reem. And I wanted to point out that we're pretty darn fortunate here in Lethbridge to have the Lethbridge Research Center and all of the scientists, and pretty exciting that there's a bunch of new scientists coming on. And it's Jean, is that right?
John, yes.
John? She was just saying Jean. Sounded like you're French. So Jean is also a new scientist. I'm not sure if you'd like to introduce yourself, and I'll turn Reem off so we don't listen to her.
Saying I don't know, another confusing diagnosis.
I took the wrong one. Yeah.
So actually, I'm from Edmonton originally, but I did spend my last 2 years in Cambridge in the UK. So yeah, I'm the new, I guess, serial biotechnologist in the enabling technologies group, and so I'm actually way upstream of what's going on today. So it's mostly molecular biology, genomics type research. I guess what I see is that when I see on the last plot taking 10 to 13 years from across going into the field is sort of what we're hearing about population growth and climate change. We probably don't have that long, so we have actually new technologies coming along that should hopefully speed that up quite a bit. So that's my job to help facilitate that. So if you have any questions, I can maybe tell you what some of those technologies are and hopefully how we can apply them to wheat, barley, probably all of our crops. So, and it's good to be back in Canada and actually in Alberta. So it's kind of full circle for me. So I'm happy to be here.
Great.
Can I ask you what kind of technology do you mean rather than breeding and training?
So what we're really excited about right now is this the gene editing technology. So it will allow us to do precisely to make changes to certain pathways in that actually, so a lot, I think all this stuff has probably come through this green revolution of irradiating and chemically mutating plants, which then takes decades to sort out and to bring some of those mutations together. But now we have the ability to precisely make changes where we want. So, yeah.
Well, thanks, Jean. Welcome. And we have one more new introduction to put you on the spot, Charles.
I was talking to him, I'm sorry.
I know, you're a real disruptor here, aren't you?
That was
We
my
love you, though.
excuse too.
Just introduce yourself and your position.
Okay, good afternoon or morning or whatever it is. So, yeah, my name is Charles Geddes. I'm the new weed scientist here with Ag Canada. As most of you know, Bob Blackshaw is kind of on his way out, so he's retiring, and I'm going to be trying to fill his position. So my research program is based on weed management, but more specifically, my experience comes from looking at weed ecology and biology and sort of integrating that into how we can manage herbicide resistance. So I foresee a lot of my program focusing on preventing and mitigating and managing herbicide-resistant weeds. So I'm brand new. I've been here for 2 weeks. So if anyone wants to come and introduce themselves, I'd be happy to give you a chat. So thank you very much.
Welcome, Charles, too. So we have exactly 12 minutes to make it for lunch. So